Hello everyone, and thank you for watching another video on qualitative and quantitative analysis of pharmaceutical excipient. Today I'm going to talk about hydrolonic acid. And this slideshow is beneficial for chemists who are doing reverse engineering of pharmaceutical or cosmetic products. And I'd like to get some information about how to do the quantitation of this excipient in their formulation. Hydrolonic acid is a natural gel and it is produced by our body, and it is found in the connective tissues, skin and eyes, and its main function is to lubricate and moisturize. Hydrolonic acid is a linear polysaccharide and consists of repeating disaccharide units. Hydrolonic acid in our body has a molecular weight of about 7 million Dalton, which translates to 20,000 disaccharide. This excipient is used in high-end facial creams, and the excipient is quite expensive. There is no USP monograph for this excipient, but there is one in EP, and it is for sodium salt of hydrolonic acid. There is an assay requirement of 95 to 105%, and also intrinsic viscosity of 90% to 120% of label value. This excipient comes in many different grades based on its molecular weight, and they go on the name of high viscosity or high molecular weight, low molecular weight, or medium molecular weight. There are at least 10 different tests in EP for this excipient, but these are all intended for raw material testing, and almost none of them are applied or can be used for determination of this excipient in the formulation. In FDA IID database, there are two entries. One is for topical gels and lotion with allowable limit of 0.01% weight per weight, and the other one is for injectables of 2.3% weight per weight. So we have two scenarios of very low amount in the gels and lotion and relatively high amount in injectables. And so depending on the formulation that you're working on, you could be extremely challenged if you are dealing with gels and lotion, or moderate challenge if you're doing injectables or something that has high concentration of the hydrolonic acid. For quantitative and qualitative analysis, I will talk about the size exclusion and reverse phase chromatography. And equipment is HPLC with UV, RI, or ELC detector. There is a published method by Kofrani and Kofrani group. They look at two different size exclusion column, Ultragel 1000 and Biosept, and it is isocratic method, and they are using phosphate buffer at pH 7. And the conclusion of their paper is that the Biosept column is acting much better than Ultragel. These are the conditions that they used for the quantitation, but I'd like to draw your attention to sample concentration that is 2 milligram per ml, and this sample was an ophthalmic solution. This method is suitable for dosage form that you have high concentration of sodium hydrolonic acid, but rather you will be challenged if you want to use it for creams and lotion where you have very low amount. So this is probably not be suitable for creams and lotion. And if you want to achieve this sample solution concentration by dissolving more of a cream and a lotion, you may have major interference from other excipients. The second method is a reverse phase chromatography with the UV detector. Approach is similar to previous method of using phosphate at pH 7. Column here is a mixed mode reverse phase column, and these columns have hydrophobic and hydrophilic stationary phase, and detection is done at 200 nanometer with the gradient method. I would like to draw your attention to the sample concentration of 0.01 milligram per ml, and this is much lower than previous method, and this method can be used for detection of hyaluronic acid in creams and lotion. And this chromatography that we see here is that detection of the sodium hyaluronate in lotion product. The hyaluronic acid elutes around 1 minute, which very much has no retention on this column, and all other excipient will be retained in the column. So that's the approach of creating a condition that hyaluronic acid elutes very fast and everything else will retain in the column, that you will get the separation. And the peak is very nice, and with this chromatography one can do the amount determination. The references are EP and suppliers CFAs and also the Kofrani paper. Thank you very much for watching this video. I appreciate if you like this video, push a like button, and if you like to see more videos from this channel, you could subscribe. But I appreciate if you have any experience working with hyaluronic acid, you could write a comment below, and I definitely will reply to you, and I appreciate your contribution. Thank you very much and have a good day.